pleasure I get to introduce Dr. Sharon Strauss, our seminar speaker for the Ecology Center series this week. Sharon is at the University of California, Davis, and I just hear she's bought an old farm in Fort Bragg now, so they might not see her too much in Davis from here on. She's an evolutionary ecologist with pretty broad interests, as you'll see over these next 2 days. She focuses primarily on plant communities, but interactions with soil microbes, with herbivores, with pollinators, and a few other odds and ends frequently come into that work. I'm not going to go through the metrics that you often hear listed when we bring in distinguished people. I'll just simply state that, yes, she publishes often and in all the right places, and she has in this political season, a huge number of citations. I don't really like lists. I like stories because I'm from the South, so I'm going to take that approach. I first met Sharon in, I believe it was 2008, in a symposium in Palma de Mallorca, so out in the Mediterranean Sea. Wasn't a bad place. Now, Sharon was there because she's an extremely important and influential scientist, well worth flying in from California. I was there simply because I was already in Spain and they could get me there really cheaply. So I haven't known her that long, but I've actually been aware of her and her work for quite some time. The first time I saw her was in 1991 at the meetings of the American Society of Naturalists, where she was given, fresh out of graduate school, a keynote address based on winning in 1990 the American Society of Naturalists Young Investigator Award based on some of her research. And then the last time I saw her, since she was too smart to go to Fort Lauderdale in August, she didn't make it to ESA this year. I was, once again, stupid enough to be there. Okay? But the last time I saw her in the 2015 ESA meetings, okay, she was being honored for having been elected as a fellow of the Ecological Society of America. So I think these kind of bookends on this career represent the importance of the work she's done and the esteem that her colleagues hold her in. The only problem with this story is that second bookend implies the end of a career. Okay, and I don't think that's happening, although once I heard about that farm in Fort Bragg, I'm not so sure anymore. But I think she's going to continue for a long time to come. So help me welcome Sharon.
Thank you very much, Gino. Your southern hospitality is showing. And it's been great seeing familiar faces here and talking with people all day today. We really had a great time, even got to sneak in a hike up a canyon, so that was pretty awesome. And gosh, I hope I can possibly live up to that introduction with my talk. I'm going to have to sit over here because I didn't bring my mouse, and we're mouse free, or pointer free. So what I want to talk to you today about is sort of related to my general interest in the interactions and the inextricable relationships between evolution and ecology, which when I first started doing my research, people would talk about ecological time and evolutionary time, and we're no longer doing that. We're sort of agreeing that evolution can happen really rapidly, and that with things like eco-evolutionary dynamics, even rapid evolution can influence current ecology really quickly right now. I'm interested in all the ways in which ecology can be influenced by evolution at many different scales. And so we can think about whether knowing the evolutionary history of a species or a set of species or their interaction can provide us insights into their current ecology. So we can think about short-term effects of history, and those might include parental effects, things that are passed on from parents to offspring that are not necessarily genetic changes, but programming changes, epigenetic changes that might alter how a particular individual responds to an environment based on its parents' experiences, and that would be one kind of history. And again, as I just spoke about recently, strong selection that rapidly changes the composition of genotypes that themselves might have different ecological properties, might result in populations that are doing different things ecologically, even over a single generation. And we call this ecoevolutionary dynamics. But also long-term history, that is the deep history of species, the speciation process, and the long-term evolutionary constraints that species must live with based on their past history, like we will never fly because we don't have hollow bones and feathers. We have some constraints that will prevent us from flying up to the top of the canopy. When a bear is there, we could run, we could try and climb, but we'll not be able to fly. And that's because a long time ago, we branched off from birds. long time ago. They were before us, actually. But, and we have now got that constraint that's basically locked into our biology. And similarly, there are all species may reflect those kinds of constraints, and how important are they in determining the current ecological roles of species and how communities then are put together with these different species. And so that's really what I'm going to be focusing on today and also tomorrow in my talk, but with taking really different approaches across these two different studies. So in some ways, I feel like coexistence in closest relatives lies at that nexus where we think ecology and evolution are the most interwoven and or maybe even prominent. So here are two sets of congeners that occur at Bodega Bay at my study site. So you can see that they look very similar and they often occur in very similar types of habitat. And what are the implications of that for how we understand ecology of systems? So Darwin had ideas about this in The Origin of Species, in which he wrote, "As the species of the same genus usually have, though by no means invariably, much similarity in habits and constitution, and always in structure, the struggle will generally be more severe between them if they come into competition with each other than between the species of distinct genera." So let's unpack this observation a little bit. So if we think about the conflicting effects of ecological similarity that close relatives might have, we have, as the species of the same genus usually have much similarity in habits and constitution, that would suggest that you're going to find them in the same types of habitats and communities because they like the same kinds of conditions. But then on the other hand, the other half says the struggle will generally be more severe between them if they come into competition with each other than between species of distinct genera. So the whole idea of like Gauss's competitive exclusion principle, the idea that if you're too ecologically similar, you can't coexist. So Darwin was suggesting that these species, because they're so similar, will not be able to coexist together. And so the question really is, does phylogenetic relatedness, the evolutionary relatedness of species, predict their ecological similarity, as Darwin asserts? And how does that affect subsequent community composition? And can we make any predictions about which kinds of species we would see together versus not? So some of these ideas have been tested with a sort of newish field of community phylogenetics, and that takes a fairly descriptive approach. So what you might do is go to a set of communities. So here we have 6 different communities, and each of them have 3 species in them. And so if we were just ecologists and we didn't care about about evolution, which many ecologists didn't used to care, and some still don't care, we might just say, oh, there are 3 species in each of these communities, therefore they're more or less equivalent. But if we start thinking about their evolutionary history, we might find that actually in this, and this is where I'm missing my pointer, but I can use the mouse. So over here, in this community, and in fact in all of these communities, they're very close relatives of each other on this phylogenetic tree. In contrast, on this community, what we see is that there's one species from each of these branches in each of these communities. So we might end up with a different type of function in these different communities. And if we were interested in sort of trying to explain coexistence or co-occurrence as a function of the evolutionary relatedness, what we would see in this set of communities is that you're more likely to coexist if you're closely related, as in here, and here you're more likely to coexist if you're distantly related. So what is it? This is exactly the duality that Darwin was talking about. So the problem with using descriptive approaches is that there are a number of caveats that you have to take when you do that kind of comparison. So first of all, we may already have had, we may have ghosts of assembly past, if you will. Maybe there were close relatives that got excluded that we don't see anymore, and that are maybe excluded due to habitat partitioning. Or maybe even more importantly, close relatives might be absent from an area just because speciation is often allopatric, often has to happen in separate habitats with a big geographic range preventing gene flow. And so what we often have is that the closest relatives are actually not close together in the same types of communities or areas or biogeographic areas. And so what we might find across these descriptive studies is that we have these, yes, communities are assembled by closer relatives, no, they're assembled by more distant relatives, but really we're not ever calibrating our communities. We're comparing, in some sense, apples and oranges when we look at what's the extant community. Does it have close relatives in it or doesn't it have close relatives in it? And that's an important thing to know before we want to jump to conclusions about whether or not close relatives co-occur or not. So what we've tried to do in this study is take a wholly different kind of approach, which is more experimental. And in that sense, we might be able to override at least a few of the ghosts of assembly past, not the really big ones about the biogeographic origins of species, but at least if they're partitioning different habitats across a relatively local area, we would be able to surmount that. And what's really great is if we can use sessile organisms because we can move them into another species niche, and then we can ask them how they do there. They'll stay there, which animals you can't do that with. So specifically, we planted species into niches of other species that varied in their phylogenetic relatedness, sorry for the typo, to the planted species, and then we interviewed them. So we said, how did you like living with that relative? And this is how we started trying to address the question of whether phylogenetic similarity really does predict ecological similarity or relatedness. And so, first of all, I have to say this work was done with my former postdoc, Brian Anaker, who's now in Boulder running the open space science program. And we've been working out at the Bodega Marine Reserve in coastal California. So the Bodega Marine Reserve is not a huge reserve. So if we look at this distance here, it's about a mile here. But we have a lot of different habitats here. We've got sort of these wetlands, and we have a little ravine. Then we have the coastal bluffs here, which get a lot more salt spray. And then we have grassland. And then the San Andreas Fault actually runs right through the reserve. And on this brown side is all sand dune, and they're on different plates. And then this is sort of this coastal grassland. So we have a really big disparity in habitat types right here that basically bisects most of the reserve. So what we did was to ask the question, are more closely related species more ecologically similar? We took a total of 16 focal species, and we planted each of them into 4 different site types. Either another site where conspecifics were there, so the focal species goes into a conspecific patch, a congener patch, so same genus, a confamilial patch, same family, and then a distant relative patch, which is a distant relative that is more distantly related than family. And these were all chosen at random from the species list at Bodega of native species. So we only did native species into native species patches. And we tried to have a diversity of families so that we might take into account the diversity of kinds of reserves. So some of the ghosts that might be implicit in this design is that, in fact, we could only use native species that had congeners, for example, at the reserve. And why are there congeners there? Well, that's another question. Why do some species have congeners and others don't? That's another whole issue that we can't address here. So we are limited to inference from other species that have congeners at the reserve. So I just want to show you a few examples of some of our species. So here we have 2 Senicula species. These are in the carrot family. And this is Daucus, which is Pusillus, which is a widespread little annual thing that grows. So this guy grows in like wooded area. This guy grows up in the rocky bluffs. This guy grows in the grasslands. And then Cirsium is growing here on the coastal bluffs also. Here are 2, this is Camisoniopsis and Camisoniopsis with Camisonia and then Erigeron. So these are the kinds of transplants that we did. We defined the niche as basically a patch where the focal species was dense or where the target resident species was dense. And we tested and we show that these patches are way denser than on average where you find them just spread across the reserve. So while the treatments were assigned with ranks, we actually were interested in treating evolutionary divergence as a continuous variable because we know that certain taxonomists don't necessarily agree with across groups. A congener in Trifolium might be less diverged than congeners in Lasthenia, for example. And so we use these 4 genes and generated a tree where we could use changes in substitutions and a molecular clock to estimate times of divergence across all of these different diverse species. So then we germinated them in trays, but we really planted them out as tiny little seedlings into our field sites. So here's just an example of, for these species, where we would be planting them. This is the grassland area, and then here we are in that dune that I was telling you about. And so we would plant them into the focal species into the sites of their different relatives. So our a priori predictions might be that if abiotic factors of site were really important, then the performance of the species would be higher when they were in a close relative site than in a distant relative site because they share the same types of habitat preferences. So that would be that part of Darwin's idea, that they are similar in habit. Alternatively, if we think interactions are more important in how these plants are performing, maybe we think that competition is going to be much stronger between similar species and that their performance will actually increase as their relatedness to the species becomes smaller, less, or as in this case, it's like the phylogenetic divergence of the species. So to understand the role of biotic interactions in this performance, what we did was for half of the replicates, we actually removed neighbors again from a very small area around these plants in order to try and assess the differences between those 2 types of effects that might be related to their performance. And then we analyzed these data where we looked at survival and growth separately as a function of how diverged the focal species was from the resident species, whether or not we removed neighbors, an interaction between that term, and then we had random effects of species, site within species, because we replicated sites across the reserve for each of these treatments, and then we did this across 2 different years. So here's our first set of results. So if we look at survival across all the sites, and this is where there's no neighbor removal, so this would be like the natural community, what we see is that a significant decline in survival that's mostly determined by a big decline at relatives that are really distantly related, beyond family level. When we reduce neighbors, we actually get a really different pattern here, where we get almost like a decline and then an increase. And in fact, the performance at distant relative sites was about indistinguishable from the performance at conspecific sites. And so we were interested in where do we see this sort of inflection. And so we actually did separate analyses where we just cut off our phylogenetic distance here at 63 million years, which is actually almost exactly the amount of divergence time between the focal species and the most distantly related, or the most closely related confamilial, so distantly related confamilials. So basically, this is subsuming up to the confamilial treatment. And then the distant is actually more diverged than family. So what we see here is that this uptick is really happening at the most distant relatives, and that we see a consistent decline from conspecific to confamilial habitats in both the presence and the absence of neighbors. Not only that, but I said that we did this across 2 years, and just to show you that even though the mean survival was very different across our 2 years, we get very similar patterns. Sometimes we even didn't use exactly the same species, but had to, because we were short on seed, substituted other ones. So we have this, 14 different families showing this fairly consistent pattern across 2 years, a pattern that we weren't necessarily expecting to see. If we look at what's happening to growth, what we see is that patterns for biomass are similar to survival, but they're subtly different. So neighbors did not reduce performance as much in growth as they did in survival. There's less of a benefit of having a neighbor reduction. And where that occurs, it's especially strong at close relative sites. And if we just look at this pattern again up to the confamilial level, what we can see is significant linear declines in growth as species are being transplanted into sites that are more and more distantly related up until this distant relative. So another way to measure the competitive effect of neighbor is this relative interaction intensity, where we look at the biomass of the focal species in the ambient plot minus the biomass in the neighbor removal plot over the sum of those two entities. And this is sort of asking, to what degree do neighbors suppress the focal species? And so this value has a range from -1 to +1. So it's negative when there's competition, and it's positive for facilitation, and 0 is neutral. So if we look at this, we find that competitive suppression scales linearly with phylogenetic distance to the resident species. So what we see here is that plants are most suppressed by conspecifics and are still suppressed by close relatives, and they actually are almost facilitated by distant relatives. And it's even more strong when we cut this off at 63 million year. So this may be one of the reasons why we're seeing this uptick in the distant relative sites. So to summarize so far, survival in ambient communities, so with the full composition of vegetation that was there, decline linearly with phylogenetic distance separating the focal from the resident species. So that says that, in fact, we are getting some signature of plants preferring to live in the habitat, surviving better in the habitats of their closer relatives. But what we see once of those survivors, when we look at the growth of those survivors, what we see is a decline to the confamilial degree of relatedness, and then this big boost at distant relative sites. So we're actually getting equal fitness of these species at distant relative sites and at conspecific sites. If we actually looked at the competitive response, then what we see is that the more closely related species were, the more they were suppressed by their neighbors. That's very consistent with the ideas of Darwin and limiting similarity. And the curve suggests that the benefits of having facilitative distant neighbors outweigh the costs of the less optimal abiotic habitats when we're only talking about their growth after they have survived in that area. And the growth effects were much stronger than the survival effects, so they tended to dominate the whole patterns that we were seeing. So this is all fairly black box. Are there other environmental correlates that we might find that are related to niche that we could measure? So we measured soil nutrients, soil moisture, and plant productivity at each of our destination sites. So we would go there and sample the soil, and then we would clip a little area that was right next to our focal plots to measure productivity and look at how that related to the different performance across our sites. So we spent a lot of money on nutrient analysis of soils for lots of our different sites, and what we find is that actually soil moisture at Bodega is highly correlated with almost everything that we measured in our soils. So it was it's almost exactly the same as using any one of those measures. So we use soil moisture in our models, and then we also looked at productivity in relation to nutrients. And really interestingly, we were expecting to see that under high nutrient plots, we might see high productivity and that it would be highly correlated. But in fact, what you see is much looser, worse relationships of productivity with nutrients than soil moisture with nutrients. Well, if we're interested in whether or not phylogenetic relatedness predicts ecological similarity, we might want to know whether or not there's phylogenetic signal in these two measures, soil moisture and productivity. And both of them have sort of weak phylogenetic signals, suggesting that relatives share preferences in these types of habitat traits. So one of the analyses we did was actually ask, well, how does the phylogenetic distance between two species relate to the productivity distance of the habitats in which they're related? And what we actually find is sort of this curvilinear response where actually intermediate. So what this means here is that closely related species have, conspecifics have similar productivities, but as you get away from that a little bit, there's actually a greater difference in productivity between these relatives than between more really distant relatives. So scratch my head a bit about what that might mean. And one of the hypotheses that I came up with was, well, maybe this is about habitat partitioning. If you have close relatives and they can't live in the same habitat, then if they've been selected to actually occupy different habitats, then we might expect closer relatives to be more divergent in the productivity of their habitats than like an average distant relative. So, and it could be that close relatives habitat partition for a number of reasons, and it doesn't necessarily just have to be about competition. For example, close relatives might interfere with each other through pollination by having the wrong species of pollen deposited on their stigmas that might reduce their seed set. If we think that close relatives that look a lot like each other, they're actually more likely to interfere with each other reproductively than species that look really different from one another. So there could be a number of other reasons why selection might act to separate close relatives that are beyond just competition. So what can we take from our results, and how can we then ask, are they at all predictive of what we see in the community at the Bodega Marine Lab? And so predictions would be that we know that species have the highest fitness at conspecific sites and at distant relative sites. So it's possible, and again, there's possible habitat partitioning among close relatives based on that productivity distant result. So these results would predict that close relatives should not co-occur very locally. That they might co-occur at Bodega, but maybe they're in different habitats, and they shouldn't be in small quadrats. So we wanted to test these results against the whole native flora at Bodega. We stuck to forbs, and so we didn't deal with grasses, partly because they're also hard to identify when they aren't flowering in our little plots. So, but basically we went and we looked at all of the native forbs growing at Bodega Bay in our little plots, and then we asked whether or not we saw patterns of co-occurrence that were consistent with our results. And we don't have phylogenies or genes that were measured for all of these guys, so instead we had to resort to ranks, congener, confamilial, distant relatives. But just to convince you that that's okay, we went back to our own results and we analyzed them by rank instead of by continuous phylogenetic distance. And what you see here is that the rank results recapitulate the results that we find with continuous phylogenetic distance. So that suggests to us that we could proceed. So we used the community composition of plots measured at all of our 51 sites, and just to make sure we were really not biased at all, we removed all of our focal species from all of our analyses because we had chosen those plots based on our focal species. So here's a 1/4 meter square plot, and inside this plot are 20 species from 19 different genera. So what we did for each of these plots was we calculated the probability of encountering a specific rank in a quadrat, and also what is the probability of your spatial co-occurrence of a particular rank as you move with increasing spatial distance from a focal plot. So what we found is that rates of co-occurrence with congeners at these local scales were very low and were less common than expected by chance based on the number of congeners at the reserve. So congeners are not co-occurring locally, which was one of the predictions we made based on the strength of competition and also the habitat productivity divergence. The probability of encountering a species of a specific rank as a function of spatial distance is also supporting our results. So what we find here is that actually as you increase the distance from a particular plot, the likelihood that you encounter a congener actually increases. It's not a huge slope, we will concede, but it is a significant slope. So what happens is the likelihood of moving away from your patch is a decrease in the likelihood of encountering a conspecific. We know that plants are patchy, so that totally makes sense, that as you move away from a focal patch, as you move away from a conspecific, you're less likely to find another conspecific. But what we see is a little bit of this spatial repulsion at this level of congeners and confamilials, which is what we predicted based on performance in our experimental transfers. And again, with distant, and of course, now we're using up all our degrees of freedom too, but again, we see this the probability of co-occurring with distant relatives is higher, closer to that focal patch. So these are completely consistent with our curvilinear responses of performance across these same areas, where performance was declining here in that congener and confamilial area and then increasing at both ends. So the conclusions are that phylogenetic distance does predict performance across niches, and that biotic interactions might be involved in the conserved niche signal to the family level. And just to throw out some other ideas, for example, if we think soil mutualists or even soil pathogens are important, and herbivores, we often know that these associations are conserved to the family level. We even have a term for herbivores, which we call oligophagous, which often refers to herbivores that eat only plants in the same family, for example. So we know that there's often phylogenetic conservatism in relationships with some of these different types of interactors that may be contributing to this niche signal as phylogenetic distance increases up to a point and then kind of goes away at the distant site. So can any of these insights be at all useful? So if we think that more closely related species are more similar ecologically, but that this niche conservatism can either promote or inhibit coexistence depending on the relative importance of the abiotic preferences and the competition or interaction outcomes, then can we use this information in any useful way? So I might argue that, for example, I heard a talk at ESA where they were talking about the diversity of prairie restorations, and the source of seed is typically grasses and composites. Like those are way more overrepresented as seed sources for restoration practices than the whole diversity of species that are actually in prairies. And this type of underrepresentation of phylogenetic diversity, of deep history diversity, may ultimately compromise the resilience of restorations if different species have different ecological niches and are performing slightly differently across space or microhabitat differences. The other way in which I was hoping to address this is to see whether or not we can use phylogenies to predict population dynamics or response to change across a number of species. So do closely related species co-vary more in their population, in their abundances through time and in their population dynamics? And by this, what I'm really interested in is if you have a species going like this over time, is their congener also more likely to be going like this over time, or is their congener going like this over time? And I think that if we find anything like that, that would also be a useful thing if we're interested in predicting, for example, how are these communities going to respond to climate change? Well, we know this species goes up. Maybe we can extrapolate that their close relatives would also increase with climate change, whereas other whole clades would decrease with climate change. Or even if we were unable to monitor some species, could we use close relatives as some surrogates? So this means we need a long-term database of a large diversity of species. We were hoping to use the Breeding Bird Survey. This is my collaborator, Marge Weber. But for various reasons, we're thinking that that might not be the best database. So we're now moving on to some annual plant databases that Larry Venable and Jenny Gremmer, my new colleague at Davis, have from Arizona plant communities. But questions could be, do common and rare species behave similarly? Is something about being rare, does that because they're actually not expressing the same kind of niche as their common relative? Or is there something else going on? And are species in the same habitat more likely to co-vary positively or negatively? If congeners are living in different habitats, maybe we don't expect correlations, but maybe when they are living in the same habitat, we do. So these are the kinds of questions that I think our results might ultimately inform and potentially be useful. So we see some signature of long-term evolutionary history in current ecology. The signature attenuates with time since divergence, and in our study, it attenuated at around 63 million years divergence. I will say there are other studies out there also showing a nonlinear phylogenetic signal, and also occurring right around that same breakpoint. So I think that our study is not going to end up being just, well, that's what's happening at Bodega. I think there's some intimations out there that we are looking at a real phenomenon. But we still need to know when shared responses and more strong interactions result in net positive versus net negative associations in natural communities. So it seems a little short, but I'm just going to, this is, the next talk that I'm going to be giving is, and now for something completely different, where I'm interested in asking some of these questions about evolutionary history and the influence on ecological niche. But instead of looking at a whole community with lots of diverse species, I'm actually looking at a whole clade where we have almost complete taxon sampling of these native mustards that occur mostly in the California floristic province, but there are at least one species over here, I'm pretty sure. Asking questions about the role of long-term evolutionary history in determining the current ecological niche in these species, and that's what I'll be talking about tomorrow. So, thank you very much. Yeah.
When you looked at the confamiliar distances, did you simply take the full data set and chop off the right-hand side, or was there?
We actually looked at the inflection point of the curve.
So all the data points that were to the left of that inflection point on the full data set were what constituted the shorter time period, or shorter phylogenetic distance gaps that were presented.
Yes. And we had the little dashed line in the full figure just so you could see where that was. Yeah. Yeah.
So you were talking about growth and survival on your curves?
Yes.
Do you think that you would see the same pattern if you looked at seed production? Or is that incorporated into
So we didn't measure seed production in this study. We basically harvested them at flowering, and we figured that biomass at flowering for annuals is pretty good. And most of these were annuals. They weren't all annuals, but so mostly I think we were worried that we had quite a bit of mortality and we wanted to have some data. By the end of the experiment, we were worried that we were running out of juice, or sample size, power. So we decided to harvest at that point. So I will say that the congeners that buck the trend are trifolium. So in those quarter-meter square quadrat, we can find 5 trifolium species. And that was shocking to me. I was like, okay, what is wrong? What is different about trifolium? And these ones we are now following up on using similar approaches, but also doing a bunch of frequency or density-dependent experiments where we invade them at different densities. We're basically following them through to fitness, so we'll have complete seed set for those. Yeah. We hypothesize that it might be because they have rhizobial mutualus and might be actually sharing mutualus, and that might be increasing co-occurrence. But there may be other wrinkles, like, for example, more and more I'm believing that reproductive interference is a very important part of coexistence that we tend to ignore. It could be that a lot of these trifolium are selfers, and so they don't interfere with each other's pollination, and that might allow them to occur in close proximity and allow them to cohabit, where outcrossing species that mess with each other's pollination might not be able to do that. So interestingly, the Camusonias also co-occur at the dunes, very in close proximity, and are also selfing. So I'm starting to build a data set of co-occurrence and reproductive system, and I'm thinking that that might end up being a more important part of the story than we think, maybe even more important than competition. So, yeah.
So, in kind of reconstructing the evolution of these lineages, I was trying to figure out like what the physical measurements say about sort of that evolution. Does this suggest that family level divergence is along some moisture gradient? Is that, or am I interpreting that right? Or is, you know what I mean?
So we found overall phylogenetic I mean, this isn't a great type of data set to look for phylogenetic conservatism, frankly. We're mostly concerned that it might be confounding our results if there were strong conservatism, but there isn't. So if you really want to look at conservatism of something like abiotic traits, you really need to do more of the clade approach that we've been using in the Streptanthus. Because these guys, what we've got are tips, and then we're excising huge chunks of the phylogeny because they're not represented in our sample, right? And that's another problem with community phylogenetics generally. It's like we can get some estimate at the tips, but after that we lose all sorts of resolution because we don't have a full representation of the tree at all, and by just cutting out all those chunks, we're missing out all the pathways along which these traits might evolve. So mostly we were just asking, well, is the signal that we see here because we see signal in productivity or signal in moisture or something like that would follow the same gradient? And what we see is a weak signal, a little bit of something there, but I don't know if that answered your question.
So it's mostly the traits that aren't captured by those gradients, I guess.
Yes, I think that's right, yeah. I mean, so this is where I differ from many, many of my colleagues, which is that a lot of people are into measuring functional traits in plants and like specific leaf area or root length or whatever. I know that those traits are really important to the functioning of plants, but I also feel that there's lots of ways to skin a cat. You can be resistant to herbivore because you're cryptic, your leaves match the background, because you're toxic, because you're spiny. And I challenge you to find a trait across a diverse group of species that would capture that defense, that diversity of defense. You can't do it because different groups are solving that problem in different ways. And so any trait that we can measure across a huge diversity of plants, like specific leaf area, do we really have any confidence that that's telling us about the niches that these guys can occupy? Yes, but maybe no. And so for me, the reason I like phylogenetic relatedness is I feel that that captures, it's a proxy for all the traits that you could measure and their intercorrelations and constraints. And that's why I like it. But a lot of people hate it because I can't tell you that it's root length or leaf area or some other mechanism that's separating them on the niche axis. And maybe it would work for congeners, but so again, I'm in the minority there. Most people would hate my approach. Yeah.
So it's really amazing and interesting that these signals do come out in the data sets, but it's also, I guess, equally interesting that there's a lot of noise there.
Yeah.
So what should we think about that? I mean, do you think then that these are strong organizing principles for community assembly, and is a lot of that noise in the data set that these are very difficult experiments to do, and is that what's...
All of the above, I think, right. I mean, this is a field experiment. Field experiments are noisy, right? The deer poops in one patch and doesn't poop and somebody takes a bite. We have recorded herbivory across our patches too, so neighbor removal.
Well, experiments are noisy, but so is nature.
And so is nature, and the fact that those...
So which should we put most emphasis on, the signal or all the noise around the signal, and trying to evaluate the importance of the principle that Darwin...
Right.
Yeah.
Well, I guess, so what I was hoping was, okay, we have this sort of somewhat noisy result. does it give us any predictive power about what we see? And so when we looked at all the other species that weren't our focal species, we did see that they predicted the associations at small scales to some degree. That congeners were repelled spatially and confamilials were, but not distant. And those were with all different species. None of these experimental species were included in that analysis. So that to me suggests that, but those regressions were huge with lots of noise and only explained a small amount of the variance of what you'd predict. But lots of other things are factoring into that too, seed dispersal, other things. But to me that said, yeah, you're onto something, because at least you're predicting these things in the right framework, which I think a lot of coexistence theory, often exceptions are in this room, but don't go back out to the native communities and ask, well, if we predict these species to coexist, what are they actually doing out there? Are they coexisting or not, right? Of course, coexistence is another, co-occurrence is not necessarily equal coexistence, so then there's that whole can of worms too, but. Yeah.
In the transplant experiment at the beginning, you used the existing plants, except for where you did the clearing, you used the existing plants as competitors.
That's right.
And it seems extremely likely that, I know you measured the abiotic variable soil moisture, the nutrients and so forth, but it seems quite likely that the average magnitude of competition is going to co-vary with habitat.
Well, that's why we measured the productivity too, in part. But I don't think, it's not clear to me that why it would be biased with the even if they're, for example, some of them are in denser habitats and some of them are in less dense habitats, but the conspecific treatment is covering across all those habitats, right? I'm not sure it's I agree with you there's variation in density, but I don't think it should be biased with respect to our treatment. I guess that's what I'm trying to say. That's part of all that noise?
Yeah.
Yeah, that's part of all that noise. Yeah. Yeah.
How did you account for the disturbance value, I guess, of like transplanting the plants?
Mm-hmm.
A transplanted plant is more likely to face mortality than a pre-existing established plant.
Yeah.
How did you kind of account for those differences?
Well, so we did transplant them like at the cotyledon stage, so they're really pretty small when we put them out there, because again, we know that microbes are important in other things, so we really didn't want them to have a very long. What we needed to do is make sure we had a sufficient sample size of equal-aged plants, so that's why we germinated, and we germinated them in sterilized potting soil. We tried to keep it as neutral as we could. It's and then put them out really young. So that was, and so when you're putting out a plant that's this big, you're really not disturbing. You would never be able to see any disturbance from when we planted, like in the ambient plots.
And there's similar species that was already existing was also the same size?
Some of them were. It depended on the species and their phenology. Not every species out there germinates at the same time. And so depending on the composition of the patches, they weren't exactly, at the same stage. But they were still young plants. We did this early in the season, so they were still young. Trying to time it more or less right. We did, there were cases where some of our target species germinated later, which they did naturally. A lot of them are cued to day length. Those guys just went out later into the habitats. So. But then their own people are also germinating later, right? So at least with the conspecific, they're kind of in the same boat. And if they land in another patch where all those other guys are bigger and there's some conservatism in when they germinate, then that's all part of the treatment. So, yeah. Oh, there's one.
You plant with the experiment, you plant the species to another place, right?
Yes.
My question is kind of stupid, and I think
That's a good question. So the question is, am I introducing invasive species and destroying the natural balance? So all of these were native to this area, so they all occur naturally there. So, and it's a small area, so we feel like we're probably in seed dispersal range across most of our habitats naturally. Like a mouse will move those seeds when they poop, a ways, and wind and other things, earthworm, whatever moves these things. And so it probably doesn't happen often in the distant habitats, but it probably still can happen over millennia that these plants are growing. So we're really not disturbing the balance because these are all co-occurring here anyway. It's more like just putting them in different habitats. What would maybe happen is that a seed would get there, but it would be the wrong habitat, and either it would germinate at the wrong time or it wouldn't survive because it would get moldy because it was too wet or something like that. And so actually that's part of the niche, which I'm sure is super important that we didn't quantify, which is the germination niche. And it's super important for so many plants, but it wasn't. I think we did put out seeds one year, and we do see some conservatism in germination across that. We did do that actually in the field. But then we rapidly ran out of sample size because we had a lot of non-germinants or a lot of mortality that we didn't pick up. And so, but we did actually see conservatism in the niche with no uptick. With no uptick, yeah. A straight line. But it wasn't, yeah. Okay, thank you.
Our introducer left.